Welcome back, everyone. I hope everybody had a wonderful break. Before I introduce the next speaker, I would like to remind everyone that if you want to ask a question, you have to raise your hand, wait for microphone to appear, and then introduce yourself. I also would like to notice that last time we only got questions from ladies and gentlemen on the left. So I would like to invite those of you who are sitting on the right, I'm not keeping score, to listen to the talk very attentively, formulate your questions, and participate in a richer way that you did before. With that, I would like to introduce our next speaker, Professor Hans Clevers, who is a professor at Hubrecht Institute. Hans is an MD, PhD from University Utrecht, Netherlands. And in the past, he served as the president of the Royal Netherlands Academy of Art and Sciences, which means that his science is incredibly good. And I'm looking forward to hearing about his work on introducing of organoids in a study of a human disease. And of course, organoids is a very exciting system that we all can use to characterize our potential therapeutics better in preclinical models.
First of all, congratulations to Fair Journey for having this, for building this fantastic building. I briefly went yesterday. It looks beautiful. We're all envious. And also for organizing this great symposium. And we're just starting, so we'll probably get many, many great talks. So we don't make antibodies, so I won't talk about creating antibodies. We, of course, use them like almost all cell biologists. I'll talk about organoids. What you see here is the inside of a small intestine. These protrusions are called villi, and these little holes in between are called the crypts of Lieberkühn. If you've done histology in medical school, you've probably heard of these. And I'll try to convince you in the beginning of my talk that these multicolored cells are the stem cells of this amazing tissue. And why is it amazing? This is another view of the gut epithelium. You see one villus, so food passes by from left to right, long tube. Mice have about 1 million of these villi. We have 1 billion. Around the base of the villi are the crypts. There's about 8 to 10 per villus, per crypt villus unit. And the stem cells sit at the base of the crypt. They divide, as I'll show you, every day, which is very unexpected. They produce daughter cells. These daughter cells rapidly proliferate for 2 days. Then they exit the crypt, turn themselves into any of the about 10 different cell types of the gut epithelium, move up while they're now exerting their functions, and about 3 days later they arrive at the tips of the villi and they die. So this entire tissue cell renews every 5 days. Now the only other cell that I know that has a short lifetime is the neutrophil. I think any other, maybe the platelet, which one could not maybe consider a cell, but this is probably the fastest cell renewing tissue of the mammalian body. And so that intrigued us a lot. Actually, this is all very old literature. This was known in the 60s. And we were trying to hope to find the stem cells at the base of these crypts. Now, we had seen 20 years ago or so that wind is a major drive. So wind signals these are like growth factors. They have been extensively studied during development in many, many different animals like flies and worms and mice and frogs. We found that when we knocked out key components of the wind pathway, identified in my lab, TCF, transcription factors, that actually this process of this very rapid cell renewal in an adult mouse comes to an abrupt halt. So wind was a major driver of these stem cells. We now know that wind is a major driver of probably almost all stem cells in the adult body. And at the same time, we found that activating mutations in the wind pathway, this was done together with Bert Vogelstein, cause colon cancer. So on the one hand, we needed normal wind to keep this process going, this conveyor belt of cells that renew every 5 days. And on the other hand, that if you mutate the pathway, you get cancer, 2 sides of the same coin. Pat Brown helped us with microarraying, so we found out what genes were controlled by wind. It's about 200. We went through this entire list that was mostly done by Nick Barker, and we found one of the genes on that wind target gene list, LGR5, which uniquely was expressed in tiny cells at the base of the crypt. here you see them. We actually then constructed mice where they emit a green light and where we could mark the stem cells in blue at will. I won't take you through too much detail, but indeed when we mark in an adult candidate stem cell, we mark these cells in blue. They produce blue daughter cells, they proliferate for 2 days, they exit the crypt, they turn themselves into one of the 10 different cell types, then they continue moving up the flanks of the villi. They're now exposed to the contents of the lumen of the gut, probably the harshest biological environment on our planet, and they only live for another 3 days, and indeed by day 5 they arrive at the tips of the villi and they undergo apoptosis as promised. Now if we don't kill the mice for analysis after 5 days, but we wait 2 years, the ribbons are still there, so the blue cells that create these ribbons are long-lived. If we score what cell types are in one ribbon produced from one cell, we see all the cell types, all 10. So these cells are long-lived, multipotent, and by our definition, were the stem cells of the gut. Now, there was a lot of resistance because it was a dogma that stem cells rarely divide and that they are essentially defined by the fact that they rarely divide. These divide every day, which becomes important. Now, another way of doing the experiment was not turning the stem cells just blue, but we created a so-called confetti allele. This allows us to create stem cells in 4 colors, and when you make it homozygous for the geneticist, you have 10 false colors, fluorescent, and our animator, Jeroen Huibe, proposed that this should be the insides of the gut, and that looked a little bit over the top, and he agreed, but then we got the images, and these are the insides of the mice, and on a blow-up, you can see about 10 individual crypts. Eventually, there's about 15 of these stem cells. Eventually, one of them wins the competition that they have. It's a neutral competition, and you end up with 1 color. And you can see that indeed every crypt has 1 color, producing cells that in parallel bands, I can't point here, but so there's essentially a number of crypts that flank these villi, and the cells move up like zebra stripes to the tips of the villi. While we were doing experiments, we realized that these phenomena were not only playing out in the gut, but also in many other tissues, including, for instance, the hair follicles. So these mice have beautiful multicolored hairs. And the 2 guys who did this, Hugo Snippert and Laurens van der Vlier, had a little contest because Hugo believed that if a mouse would escape and he would flip off the light and then use a black light, that he could actually retrieve the mouse in the animal house. And that did work. So they are beautiful. They have these multicolored hairs. Well, this experiment essentially implied that LGR5, and that's what we now believe, is a marker probably of all stem cells of epithelia, ectodermal, mesodermal, endodermal, and always these stem cells are driven by wind in their active state. So that I think is a general rule. These rules don't work for the hematopoietic system, for instance. So bone marrow stem cells cannot be expanded outside the body of the donor, even after 40 years of trying, 60 years of trying. But I'll show you, probably holds for all internal organs. Now, I already said that we found that against dogma, these stem cells divide every day, and the dogma was there because it was believed that stem cells, when they divide, can sustain DNA damage. You can lose them, or even worse, they can be turned into cancer cells. So it was believed that stem cells very rarely divide, as they do in the bone marrow, and that only their daughter cells would then strongly amplify cell numbers. Now that is a very logical hypothesis, but it doesn't hold in the gut. We now also know it doesn't hold in the stomach, it doesn't hold in the hair follicles and some other tissues. So these cells are always proliferating. They go through about 1,000 consecutive cell divisions in the lifetime of a mouse. In humans, they probably go through 20,000 consecutive cell divisions. So it's quite amazing, the logistics of what these cells do. We don't know how they do this, but they manage. But based on this observation, Toshi Sato, a Japanese gastroenterologist who has his lab back in Tokyo now, took up the challenge of seeing if we could grow these cells. Now again, there was another dogma in the stem cell field that normal tissues cannot be grown in the lab. You can keep them alive for a brief time, but they cannot be expanded. And everything that grows in the lab is a cancer cell. And if you think about all the cell lines that you're using, they are invariably cancer cells. If you transplant them, they make a tumor or they cause a leukemia. So So many postdocs in the lab didn't want to take this project up, but Toshi did, and we sat down and we learned quite a bit about the growth factors needed to keep the crypts active in a live mouse by just randomly knocking out all sorts of signaling pathways. And we thought we probably only need 3 growth factors. We need to activate Wnt. For Wnt, we use a molecule called R-spondin, secreted molecule, easily produced, unlike the Wnt. Later, we found that it actually was the ligand of our LGR5 receptor. LGR5 is a surface 7 transmembrane receptor. EGF, strong mitogen for epithelia, and a BMP inhibitor noggin. And if we just combine 3 recombinant proteins, no serum, just standard medium, and then we use Matrigel, which is still quite magical. It's a 3D gel, liquid at 0 degrees, so easy to handle, and a gel at 37 degrees. We put 1 stem cell in, you see that here, and then we get these structures growing that are not the intended outcome, because we were hoping to create many stem cells, but rather than just creating a lump of stem cells, like one would do with iPS cells or ES cells, we got structures. And they, if you look carefully, they are mini versions of the gut epithelium. All these buds that you see growing out over a movie that plays about maybe 3 or 4 days, they are the crypts, so there are multiple stem cells. There are the pennate cells that are usually the neighbors of the stem cells, and every other cell type of the gut is there in the right numbers and in the right location. So really surprising that a stem cell on its own makes a mini version of the gut. Now, by sequencing, we never found that they have sustained oncogenic mutations, so we've had them in culture for many years, much longer than you would ever have a mouse live in an animal house. Yet, because they grow so vigorously, they grow about 10-fold per week. So in a matter of a month, you have 10,000-fold amplification of tissue, much like cell lines. So we then teamed up with Mamoru Watanabe's lab in Tokyo. And you see the names of his people here. And Toshi in Utrecht sorted 1 stem cell, and I stress this, 1 stem cell from an RFP-positive mouse, so a mouse that is red, and the stem cell is green. And he grew that 1 stem cell up in Utrecht into about 100 million cells in the form of these mini guts, as he calls them. We then sent them to Tokyo, and there they were transplanted simply by inserting them through the anuses of mice treated with DSS, which is a chemical that causes inflammatory bowel disease, very popular model for the study of inflammatory bowel disease. And over the next few hours, apparently these Dutch red organoids float around. They have their basal site on the outside, so the integrins are on the outside. They will not adhere to healthy tissue, but the moment they see collagens or laminins or similar materials, they will adhere with their integrins, open up, and like a living Band-Aid, they seal these lesions. Now, you might have noticed that this was not a real experiment that is here. So top left, you see a black Japanese colon, and these orange-red patches are the Dutch tissue that actually landed there and inserted itself into the healthy epithelium. And you can only find these patches back by confocal microscopy, by looking for RFP, for red fluorescence protein expression. And by any other means, you see a few markers on the right. This tissue is fully normal and fully integrated. So one prediction would have been these stem cells are exhausted. That's what hematological stem cell people would say, because we have expanded them dramatically. They probably can no longer divide. That's not the case. They divide happily. Telomeres, I should stress, stay very long in culture, actually get longer. And the other prediction would have been, well, somehow with all this excessive stimulation by these growth factors, you have turned these cells into cancer cells. We have never seen any neoplastic event happening, so no adenomas, polyps, or no cancers occur in these mice. And based on this, Mamoru has taken this further and currently actually is performing his first-in-man trial, trying to treat inflammatory bowel disease patients with their own organoids. So he samples small bits of normal tissue by colonoscopy, expands it 10,000-fold in culture over a month, and then is aiming to transplant this back into the same patient. So no transplant issues there, no HLA problems in this setting. We then realized, this is about 10 years ago, that of course this could be used for regenerative medicine, the way Mamoru is doing this, but there are many more applications, and I'll give you some a lot of short stories in the next 30 minutes or so. So applications in cancer are quite obvious because we grow normal tissue for long periods of time, so it should be very easy to grow cancer tissue. And that is true, so we meanwhile had learned to do this for human tissue, and you can see here a whole list of organs that we can turn into organoids. I should stress, what we do is we take a small bit of tissue. We don't need to sort out stem cells. Many organs actually have no actual stem cells. They have cells that are recruited to be a stem cell when needed, but like in the liver, I'll show you that hepatocytes can actually serve as stem cells while they stay hepatocytes. So we take small bits of tissue, small biopsy is good enough, we grind it up, put it in Matrigel, add the right cocktail of growth factors, which is different for every tissue, similar but not identical, so we always have to optimize this. It's always active Wnt, active tyrosine kinase receptor, and blocking of BMP TGF-beta. But for instance, for breast tissue, we have to add estrogen. For prostate tissue, we add testosterone, et cetera, et cetera. So that usually takes about half a year or a year to optimize. But if all is in place, then we can at least grow them exponentially for a year while they stay phenotypically and genetically normal, and they essentially have all the cell types of the original tissue. Only epithelial elements grow out, immune cells disappear, blood vessels disappear, fibroblasts disappear, nerves disappear. So they can be added back, but then you have to get them from, if it's an immune system, from the same individual, which is a bit of a challenge. But if you think about all of these organs, the essential functions of internal organs are invariably taken care of by the epithelial part of that organ. So essentially we grow the functional parts of the organs, we lose all the rest. And for cancer, for instance, carcinomas by definition are epithelial. All carcinomas are epithelial, so we can always grow them, and they account for about 90% of all cancers in adult patients. Now, we can use these organoids to model cancer and hereditary and infectious disease. And cancer, here but now many other labs have done very similar things. So essentially, one can build biobanks of cancer patients. And in Utrecht, for colon cancer that you see here, we probably have about 400 or 500 lines now in the freezer that are fully sequenced, that you can grow forever. They grow with different speeds, I should stress. And for some of them, we also have healthy tissue, if we can, so we can actually compare side by side healthy tissue and the cancer tissue. Now, you can sequence, you get beautiful sequences because the tumors only contain cancer cells, everything else is gone. Also, of course, a disadvantage in certain models. But most importantly, we have the live cancer cells, and we can now expose them in the lab to a number of drugs. And a foundation in Utrecht that we've set up together actually with Ton Lochtenberg, who speaks next, has been able to do 30,000 compound screens still in a fairly crude version of this assay. And there are now a number of companies that are trying to robotize this to make this much faster and to make it more useful for drug development or for personalized medicine. And so this is what they look like. On the left, healthy, on the right, a tumor from the same patient. And when you sequence, you can confirm actually that they have the same mutations as you'd see in the primary tumor, and they don't add more mutations or oncogenic mutations in culture. We can drug screen. Indeed, this was all done by hand, not on robots or anything. Works well. It's extremely robust as an assay. Of course, it's very important that what you measure in vitro reflects what happens in vivo with the patient. While we were trying to do these observational trials, a beautiful paper came out from a consortium in the UK that shows that in at least 4 or 5 small phase 1-2 trials, where they, at the same time that the trial started, they isolated tissue for organoids, that the predictive value, at least for the drugs in those trials, all experimental drugs, was over 85%, both in terms of sensitivity and resistance, so extremely high. You have to realize that when a patient, so first-line treatment for the average cancer patient probably is effective for 40% of patients, and 60% of patients, they get the drug, they get all the side effects, they lose time, and maybe the second line will work, maybe it won't work, but the situation is already much worse than it would have been. So this would be comparable to testing antibiotic resistance in bacteria. So when you have a pneumonia with a bacterium in your lungs, the bacteria get cultured. In the lab, it's determined what is the best antibiotic for the individual patient, and then after in my medical school days, it was 3 days. It's probably now faster by PCR, but you're then given a drug that will surely kill your bacteria. Now, for cancer, you're classified, and then it's a statistical argument why you're given a drug. You're in a group that will respond, but you don't know whether you are in the responders or the non-responders. You only find out when you get the drug. But this would offer the potential to actually determine whether your cancer cells do get killed by the drug that is designed for your group. And maybe you can shift. Well, there are now numerous papers that report similar observations. In 2019, there were several studies where we were involved. We're not the lead authors on these papers, that all come up with a similar predictive value of 80 to 90%. I should stress, though, that this is a tedious experiment. It takes 5-6 weeks, sometimes longer, to be able to read out. That's too slow to be meaningful in the clinic. Also, you need skilled personnel, the logistics of getting as you know, pathologists like to look at tissues, but the first thing they do, they put it in formalin. That's good for looking under the microscope, but it's too late to grow them as organoids. That has been a major challenge to set up the logistical pipeline to get living tissue in the lab to culture. That, I think, we have worked out. And there are now a number of companies that are built, as I said, robots that will allow this to do this in about a week, so that you don't have to expand the cells. All is done at a very small scale, and you can think of now of systems where you could do really high throughput screens of 100,000 or even 1 million compounds, given the robots that are currently being developed. Another way of doing cancer research is combining this with CRISPR, and this is 2 studies, one from my lab, Jarno Drost, the other from Toshi, who at that time was already independent in Tokyo about 5-6 years ago. So we both realized that the growth factors that we empirically had found to be required to expand these stem cells and to be necessary in a crypt, that the cancer of in this case the colon, activates or blocks exactly the same pathways as we activate or block with our growth factors, and that's here. So the most common mutation in colon cancer is the loss of APC, one of the first tumor suppressors ever cloned. 80 to 90% of colon cancer cases lose APC. This activates the Wnt pathway. We add Wnt to the medium. So the prediction would be, if we take a normal organoid and we take Wnt out of the medium, they will die. But if we mutate APC at the same time, they will live and they no longer require Wnt. Now, the same thing holds for KRAS and EGF. The same thing holds for SMAD4 and Noggin. Noggin is our BMP inhibitor. SMAD4 is a transcription factor in the BMP TGF-beta pathway. So if you knock it out, you would no longer need this. And so we both set out using CRISPR to try to recapitulate what Bert Vogelstein and Eric Fearon in I think 1990 already proposed, that there is a relatively ordered succession of mutations that take a normal colon stem cell, presumably, finally to become a full-blown invasive colon cancer cell. And yeah, there's a little movie that illustrates this again. So here you see the normal colon epithelium. Colon cancer probably takes 20 years to develop, so an early adenoma, intermediate adenoma, late adenoma, all driven by additional mutations, and finally an invasive and metastasizing carcinoma. And that's what we try to engineer. So we start from a normal colon organoid. We grow it up. You see the growth factors on the right. We knock out APC with CRISPR, leave Wnt out of the medium, everything dies, and only the APC mutants will grow out. That's what we observed. Now, next step is, I believe, P53. Yeah, so you see that here. So here we can kill the wild types with a small molecule called nutlin, and everything dies, but now we have the double mutants, APC P53. They still need the growth factors on the right. We now target KRAS. Here we have to do a knock-in, works quite easily in organoids. So we activate KRAS, leave EGF out. Now they have 3 mutations, and they only need knock-in. And the last one, you see here, we target SMAD4, transcription factor in the BMP pathway. If we knock that out, we block the BMP pathway. We leave the blocker out of the medium, and they happily grow. So now we took about 3 months. We now have a whole series of colon organoids with 1, 2, 3, or 4 mutations in all combinations. We can now actually do this in one shot for up to 6 genes, and then we get a library of using base editors. I won't talk about it here, but CRISPR base editors are really incredible reagents. You can change single bases anywhere in the genome of these cells. And you go in one shot, we can hit 6 genes, and then you get a library of organoids where every organoid will have a different combination. Some have all 6, some have 5, some have heterozygotes. And in one shot, you make a large library in the same genetic background. So if you want to study if a drug works or doesn't work, this is probably the best way and to see how that correlates to mutations. When we transplant these into immunodeficient mice, on the top right, you see the 4 mutation version. That actually is now a full-blown carcinoma. It invades, as you can see, and it metastasizes to the liver. So this essentially confirms what was known. You need 4 mutations in these 4 pathways to take a normal cell into a full-blown colon cancer cell. Liver, I'll give you just a few images. So the liver has 2 main liver cell types: cholangiocytes, which build the bile ducts, and hepatocytes, which are the chemical factories and the producers of many of the serum proteins. All other cells in the liver come from elsewhere, like Kupffer cells come from the bone marrow, stellate cells are essentially fibroblasts, blood vessels come from outside. There's 2 real liver cell types. Now, the liver is the most regenerative organ after damage of our body. You can either chronically damage the liver by toxins or by viruses. Then the cholangiocytes become oval cells, and they're really like stem cells briefly, and they can make both cell types, so that's the cholangiocytes. And the other way, first I'll show this. So here from donor livers, we sort, on the bottom, we sort cholangiocytes, a few % of the cells in the liver. On the top, hepatocytes. Hepatocytes are key in drug development for many, many different reasons, but nobody could ever produce them. You actually get them from a donor liver, and human hepatocytes. The cell lines are cancer cell lines. They really are not hepatocytes. And also iPS-based hepatocytes. They have some resemblance to real hepatocytes, but they're not there yet. Cholangiocytes were very easy to grow. As you can see on the right, very surprisingly, these are full-blown differentiated cells that transport bile, take bile up and transport it to the gallbladder and then into the duodenum. Very beautiful cells. 1-to-1, you can take a fully differentiated cholangiocyte, put it in the growth factor cocktail. It'll de-differentiate in 2 or 3 days, and then be bipotent and make both cell types. Mary Hoog, who the first author on this paper, has gone on with this in her own lab and shown that what actually happens at the genomic level. So you get a rapid demethylation, et cetera, of the DNA. You really see an in vivo physiological reprogramming of fully differentiated cells to temporarily become a stem cell. Now, it turns out that this happens in many, many other organs, and it's totally different from the dogmas from the hematopoietic field. Anyway, so these grow, and they... here you see a confocal, so there are these hollow cysts. If you put them together, they make a tube, so they really make a bile duct, and they're fully differentiated. But the hepatocyte is essentially the most important cell here, and that could never be cultured. Now, we tried lots of things, and then Hu came up with a cocktail of about 12 different growth factors and small molecules. First in mouse, and this is a mouse where the albumin gene is marked in red, as you can see here. And starting from a single hepatocyte, we can grow organoids. They look very different. They almost have no lumen. Most others have a lumen and have highly polarized cells. Then we learned how to do this for humans. Again, Hu did this, who now has a lab in China. Here you see the production of alpha-1 antitrypsin. So these are large cells, 40 micrometers, large nuclei. If you ever look at liver, big nucleoli, and you can see the amount of this serum protein is extremely high. The same for albumin. We get the same levels as a newly isolated, freshly isolated hepatocyte. You see a 1 mitosis on the right. So they divide. They don't grow very fast, maybe 2 to 3-fold per week. But they grow forever, particularly if the donor is very young or if it's a fetal liver. I think I have a few other images. Yeah, here they even do more. Yeah, it's probably better to see in the next slide. Yeah, so what you see here is these large hepatocytes. Essentially, when you do single-cell sequencing, they are hepatocytes, but some of them divide while being an hepatocyte. So they are stem cells and hepatocytes at the same time. But they also create these, you might see these little ducts, and these are the bile canaliculi. So the bile duct takes the bile out of the liver, but the hepatocytes produce the bile acids. They secrete it to the apical lumen, and the apical sides of 2 hepatocytes merge together to form a channel. And that's what you see here. So you see these little, we stain this for MRP2, which is the bile transporter. So this is indeed a fully functional hepatocyte. It's producing bile acids. It secretes this into the bile canaliculus that it creates itself with its neighbor. Then the bile ends up in this big yellow blob, which is probably where the bile duct should have been. And because there are no cholangiocytes in these organoids, that's where they accumulate, and eventually they burst open and they release the bile acid. But this shows how complete these organoids are, and how, I think I should stress, how self-organizing they are. We don't do much. We just add a few growth factors, then we let them do their biology, and they recreate, like stem cells in vivo do, they recreate the normal tissue. These can also be transplanted. This is in a popular transplant model, FAH-deficient mice. In blue, you see the mouse liver, where on normal diet, the hepatocytes slowly die. But when you transplant these hepatocytes, the human hepatocytes, they will create these growing islands, and you might appreciate these little yellow dots. They are Ki67 positive cells, so they are dividing 2 months after transplantation. So these islands keep on growing till they fill up the entire liver of these mice, and you can see milligrams of human albumin in the serum of this mice. So they really are functional. This was done in collaboration with Ipe de Jong. I have a few more stories. This is a paper that came out last year, and essentially I didn't know this project was happening in my lab. It's my 2 PhD students, Cayetano and Jens, together with Axel, PhD student in Ruben van Boxel's lab nearby. They had read about a version of E. coli, a strain of E. coli that is quite common. There was a long history of publications on this strain, which has about 60 extra kilobases in its single chromosome. You see it here on the top right, and those 60 kilobases hold about 10 genes that together constitute a synthesis pathway for a polyketide. PKS is polyketide synthesis. And there's actually many of these PKS islets in the bacterial world. And these were important, people thought, because if you take E. coli, first of all, they occur in humans. Probably about 20 or 30 people in the etiologists have this particular E. coli in their colon. And you'll learn that it's not a good idea to have them there. Because if you take them out and you culture them and you put them on the human cell line, almost immediately you see double-strand breaks. So, the product of this locus, it's called colibactin, it's a small molecule, I'll show it later, will enter human cells, so secreted from the coli, enters human cells, causes double-strand breaks in the DNA. Now, this is not what you want to have, particularly not in stem cells. So, also somewhat surprisingly, there is one strain called Nissel that is a very popular probiotic that has this pathogenicity island. So it's a genotoxic bacterium, it was already known. We'll show exactly what it do in the next slide. But it's taken by, or it's prescribed by gastroenterologists. There were many trials ongoing when we started doing these experiments around the world in serious university hospitals for inflammatory bowel disease, irritable bowel disease, psoriasis, and other chronic diseases that are often difficult to treat. So they asked the 3 PhD students, is this colibactin, is it not simply causing double-strand breaks, but does the resolution of those breaks, does it result in mutations? So can this be not only a mutagen, but can it be a carcinogen? And here you see so while we're doing this paper came out, beautiful paper in Science on the chemistry of colibactin. It's extremely labile, so I don't think any lab has ever synthesized the entire molecule, but this is the deduced structure. And you see these two AD on the 2 sides. The warheads, the 2 warheads, the left and the right side of this molecule, in this paper are proposed to covalently bind to A residues in DNA. And if this happens between 2 strands, this is an interstrand crosslink. This is not compatible with life for that cell. So that cell then has to somehow get rid of the adduct. And in doing so, it apparently causes a double-strand break. That was known. Here is our experiment. So we got from collaborators in France, we got the strain and also a mutant version of the same strain. So it's a very nicely controlled experiment on the right. So the left one is the bad one, the right one is the negative control. It cannot make colibactin. And the idea would be is that we, first of all, learn how to grow these in the lab. That turns out to be as easy as any other E. coli. Many other gram-negative anaerobics are very difficult to culture. Anyway, so we can grow them on agar plates. We inject them It's the first experiment into a normal human clonal organoid. So every cell in these organoids have exactly the same genome. They were recently cloned. We now expose them over a few days to the bad bacterium on the left and the good one on the right. And then we simply look, do we see the double-strand breaks? So that's, can we confirm what's in the literature? Now, you see in the middle, PKS plus. In blue, every dot is an organoid that was injected by hand by the PhD student, quite an experiment. And the red dots in this magnification are individual double-strand breaks that are stained by gamma H2AX. And you can see there's many, many breaks, and on the right, the negative control, in the entire culture, they saw one break. So we could confirm in organoids what was known for experiments done on HeLa cells or other human cancer cells. So then they did a heroic experiment. So now they just not injected once, but injected over 3 to 5 months in several experiments. The problem is that bacteria escape from the organoid. They will overgrow the culture. They don't need the cells. So they had to cure the organoids every Friday and then inject again on Monday. Did this for, with antibiotics. So they did this for about 3 to 5 months. And then after 3 to 5 months, we could now sequence the ones on the left whole genome and the ones on the right. And the hope, the expected outcome would be that the ones on the left have more mutations and maybe they have recognizable mutations because there is a very specific mutational process. Now, if we would just sequence the one on the left, we would not see anything because every cell might have many mutations, but it'll always differ from its neighbor. So everything gets diluted out. So they then subcloned single cells from these organoids, grew them up, just to amplify the genomes of single cells. It's like a PCR for the whole genome. And then they sequenced subclonal organoids. So you see the DNA here, whole genome sequence on these clones. And on the left, you see the bad one. On the right, you see the control. We can just ask, okay, where are the point mutations in this case? And it turns out that, of course, in culture, you will always see somatic mutations with a certain rate. But the ones on the left had, first of all, many more mutations. And also they had a unique type of mutations. And I won't take you through all the bioinformatics, but it turns out that we see a very specific, easily recognizable motif that gets mutated. A TA pair in the middle at position 0 is either changing into any other base pair or it's deleted, so just you lose. So in the second case, you have a frame shift if it's in a gene. In the first case, you'll have a missense mutation or a stop codon. And this happens in the context of an A at -3. If you now go back to the model on the top left, indeed, this looks like the proposed mechanism for colibactin, binds covalently to an A -3, covalently to an A at position 0, opposite the T. Now the cell wants to repair this, and in doing so, it takes out the base pair at position 0, or it changes it into another base. And so this is very easily found, because no other mechanism causes these types of mutations. Of course, also this was very artificial, but then we could ask, do we see this back in large cohorts of cancer patients? First, we worked with the Hartwig, where they have sequenced almost 4,000 metastases of a large variety of different cancers. And you might see this, but you see the sort of the dots that go up on the left of this graph are the colon cancers. So about 10%, we now know about 15% of colon cancers show this particular type of mutation. So the A at -3 and then the TA at 0 changed or deleted. No other cancers do this. The occasional bladder cancer has it, but E. coli can chronically live in the bladder, and the occasional head and neck cancer has it. But like breast cancers, which are sterile, never show these types of mutations. And then the second cohort from Genomics England was only colon cancers, but we again saw this mutational signature in about 10 to 15% of the patients, and mostly in the left-sided colon cancers. So this is associative evidence, but we believe that indeed in the history of some colon cancer patients, this bacterium has been present, has caused mutations. And what I then show you here, some of these single base changes actually hit cancer genes, and very often APC mutations seem to be caused by this particular mechanism. And therefore, one would conclude that these bacteria are indeed mutagenic, and the mutations they make are very easily recognizable. You see them in a subset of cancer patients. You might now think about removing this strain from the human population. They're very easily found, and like the only other bacterium that's linked to cancer is Helicobacter, or causatively linked to cancer, Helicobacter. For stomach cancer, there the removal of the bacterium works very well. So there are now a number of cohorts around the world are being followed. It's being determined who carries these bacteria. Are there only kids? Are they within families? Are there certain ethnic groups that have more of these bacteria? And people think of ways of removing these bacteria now. And I think a strong recommendation is don't prescribe these PKS positive E. coli to people that already have inflammatory bowel disease and have a high chance of undergoing cancer. Then rapidly two examples of using organoids for infectious disease. The first one, actually when we went into lockdown early last year, I should say actually I was allowed to take my mask off if you're worried, and I'm vaccinated, so that should be okay. So we realized that ACE2, that was already immediately known that ACE2 was the receptor for the virus. The same receptor was already known for the original SARS virus. ACE2 is most highly expressed on gut epithelium, we knew that. And there was also an indication that quite a number of patients don't initially present with respiratory symptoms. They present with gastrointestinal symptoms, sometimes only gastrointestinal symptoms, so nausea, diarrhea, stomach aches. And there were some scattered reports that one could detect by PCR viral RNA in stool, indicating that this might be a second organ that's infected. Maybe clinically not too relevant because you will not die of this if you end up in the ICU is because of your respiratory problems, but it might be an important route of transmission of the virus. So we teamed up with the lab in Rotterdam that already had been working on coronavirus for a long time, done a lot of work on SARS and MERS and also on SARS-CoV-2. So Mart Lamers and Bart Haagmans. In my lab, my names are not there, was Joep Beumer and Jelte van der Vaart. And we could show indeed human small intestinal or colon organoids, when given a little bit of the virus, they would amplify the virus dramatically, actually much better than airway organoids. And you can see this in the order of 10 to the 4, 10 to the 3, 10 to the 4-fold amplification in a matter of less than 3 days. And we do this both for live virus and for viral RNA. So these are really infectious particles that are being produced. Here you see after 24 hours in white, one cell that is infected in a human intestinal organoid. We stain in white for the nuclear protein of the virus, and you can see a little bit later, two days later, essentially the entire organoid is now positive for the virus. So they really infect and then propagate in culture. The cells actually do well. We do all sorts of analyses, single cell sequencing, but they don't get killed right away. Other viruses do this. So that's probably a reason why these patients are already infectious before they ever note that they are sick. So they already produce a lot of virus while there's very little pathogenic effects in the tissue yet. This is published. This actually paper just also got accepted. We then asked, can we now use this model to find out what human genes are used by this virus for the infectious cycle? And of course, ACE2, the receptor, was a clear one, but there was a whole list published, 3 cell papers with genome-wide screens, CRISPR screens, that had 50 genes that were key. So we first looked if we could actually use this system. Now with base editors, we very rapidly can knock out any gene just using stop codons. And what you see top left graph is SARS-CoV-2 needs ACE2, and in blue, the blue lines, if we knock out the receptor, they're no longer infected. So it works. SARS-CoV, bottom right of the 3 graphs, also is blocked if the receptor is no longer there. But MERS, you might have heard of this Arab version of corona, that still constantly jumps from camels to humans. As far as I know, no reports yet that it jumps from humans to humans, but it's highly lethal. But 50% of the people who get infected will die. It was also known that it has another receptor, and this one still infects, so it's a negative control. And the other receptor is DPP4, so if you knock that out, SARS-CoV-2 happily infects. SARS, I don't show it, also happily infects. But now MERS can no longer infect these organoids. So that's the positive control here. We then went through a large number of likely human genes, and you see a number of them here. I just showed that TMPRSS2 are the blue lines, the bottom 4, these are all 4 different clones, appear to be essential. Now TMPRSS2 is known to be a protease on the surface of cells, on the outside of cells, that will cleave the spike protein and then allow the virus to enter the cell. So that was another known and fully established target gene, but none of the other genes, when we knock it out, have any effect in this system. And most importantly, cathepsin L, CTSL, is a protease key in the endocytic pathway, key in virocells for infection. So virocells are African green monkey cells that are used by almost all virology labs. They're more fibroblast-like, they're not really epithelial-like. When we knock cathepsin L out, we don't block the virus, as what would happen in virocells. We essentially enhance the entry of the virus, as you can see, the purple lines on top. So we also then noted that chloroquine, which in our hands also is a great, or hydroxychloroquine, a great blocker of infection. As you know, several presidents of entire countries were very happy about this drug. And I know that many of the hospitals in Holland actually prescribed chloroquine in the beginning of the pandemic because there was nothing else and there was some convincing evidence. So it does block entry into virocells fantastically well. We now know it does nothing in patients, actually only harms the patients, and it does nothing in organoids. And the big difference here is that organoids are primary epithelial cells and virocells are fibroblasts. Now what does the virus encounter when it enters a patient's airway or intestinal tract? It encounters polarized epithelial cells. On the left, you see here how the virus enters in virocells, so it's by endocytosis. This is sensitive to chloroquine, it's dependent on cathepsin. On the right, you see what happens in a primary epithelial cell. The same receptor, ACE2, is involved, but on the primary epithelial cell, TMPRSS2 will now cleave the spike, activate spike, and the virus directly fuses with the membrane and injects its RNA into the cell. So there's no endocytosis, there's no role for the cathepsins, and also there's no effect of chloroquine. So here, one conclusion would be that actually next time, the next pandemic, maybe do the screens on virocells, but then as an intermediate step, try on primary epithelial cells because they are closer. They're not a patient, but they're closer to a patient than these green monkey cells. And then finally, just because the movie is very nice, Cryptosporidium is a malaria-related parasite. You might remember that it's a malaria is a eukaryotic parasite that goes through a mosquito and the human host and actually cycles through asexual and sexual stages, very complicated life cycle. Cryptosporidium does the same, but it only has one host, so it doesn't need insects for transmission. You see it here, but this is here in the movie. So here, Cryptosporidium is a big problem in veterinary sciences, but also in third world countries, chronic intestinal disease, no good drugs. And what we hope to do is to replicate the cell cycle, the life cycle in organoids. They could never be grown in the lab. So it starts with oocysts. So these are, you see them here. Typically, cows will pick it up from grass, from infected cattle. These oocysts, here we inject into an organoid. That's the idea of the experiment. So the oocysts will land on the epithelium. We find that they actually, the little critters that come out, the sporozoites, there's 4 of them per oocyst, that they actually infect fully differentiated enterocytes. So these are the cells on the villus that you never see in cell lines, you only see in organoids. They make type 1 neurons, so once they enter a cell, these type 1 neurons release their contents. I think there's about 8 again of these type 1 merozoites. There's an amplification step. They do this once again. So they will again infect a fully differentiated enterocyte. Again, this cannot be done in cell lines, but works well in organoids, as I'll show you. We get a type 2 neurons, again with a defined number of nuclei, so that's how we can identify them. The type 2 neurons come out. Now we enter the sexual cycle. We're about 2.5 days into the experiment. Now we make 2 types of structures. In front, it's the male version that makes microgametes. In the back, it's the female version that has 1 macrogamete. And here you see a microgamete coming out and fertilizing and forming a zygote. And then this structure will now become an oocyst and be released from the epithelium and then leaves the body with the stool and is then eaten up by another cow or occasionally by humans and causing a chronic infection. Now this works well in these organoids just by EM. we see all the stages, and after they produce very high numbers of oocysts, and when we take these oocysts, they will infect mice. So they're really infectious particles. Matthias Lutolf, then at APFL, has actually used this for a chronic infection model where he created from the minigus, he created a tube that he could maintain for months in a gel, while basically have fluid flowing through the tube. When he infects these tubes, there's a Nature paper very recently from his lab with the oocysts. You can see that they chronically infect these tubes. So this is a very nice model for chronic infection by a parasite that could never be studied in the lab or outside experimental animals. And with that, I reached the end of my seminar, and I'd like to thank you very much for your attention, and I think I left some time for Q&A. Thank you.
Wow, I would just say, wow, the advances that you describe are utterly amazing, and opportunities they open for us to study anti-cancer treatment, antiparasitics. While the audience is collecting their thoughts, I would like you to ask a question about the importance of scaffold. There is a long-standing debate in hepatology and fibrostenotic IBD whether you can take a highly cirrhotic liver, repopulate it with normally behaving cells, and reverse the disease. Your results seem to argue that these cells form normal structures in very artificial environments. So how do you feel about an opportunity to use hepatic cell transplants in a situation of cirrhotic liver or fibrostenotic IBD with an attempt to reverse the disease?
Yeah, so we've of course been thinking a lot about this. I think when you do this very late, so when the liver is really scarred and cirrhotic, it will be very, very difficult to recreate a functional liver. So there you really need a liver transplant that's currently being done. Also, that is always done very late in the disease process. There are some indications, not from our lab, that actually if the fibrotic process is already starting, you then put healthy normal cells in, they will reverse that to some extent. So if this ever becomes a therapy, it has to be earlier than where we now transplant cancer patients. There would be much less of a logistical problem because you can freeze these cells easily. So you can actually have a bank like a blood bank. You could have a bank of liver organoids and then just thaw the one out electively when you want to transplant the patient. There is actually currently, I don't think I'd mentioned it, a trial has started for a different disease, dry mouth after head and neck cancer and radiation, where the parotid gland and the submandibular glands basically get destroyed by radiation. Terrible disease, your teeth fall out, extremely painful, difficult to eat, difficult to talk. The animal experiments, so human salivary gland organoids in destroyed mouse, radiated destroyed mouse, they restore the function of these glands. And as we speak, a trial has started in Groningen in Holland, where they are now using these cultured parotid glands, I believe autologous cells, so they basically take the few cells that are still alive, expand them, and inject them through the skin into the salivary glands. And they hope to see the same thing as they've seen with human salivary gland organoids in irradiated mice. So that would say that for that particular organ, it looks like it can repair even pretty bad changes to an organ. Yeah. But this is all very early days.
Yeah. Thank you, Hans. René, can we have a microphone on the right, row 3?
First, thanks for a beautiful talk and beautiful science and very nice movies. I'm René Hoed from Montes Bioscience in Leuven, a small biotech. And sort of, intrigued by these organoids, how far you can go. For example, can you mimic the tumor microenvironment? Can you introduce T cells, macrophages? And we're particularly interested, for example, in endothelial cells. And have you looked in that? First question is, have you looked into this? And secondly, do you think it's possible in the future to really make organoids which mimicking really, the whole tumor microenvironment?
Yeah, so my first answer is that my lab actually always has gone for the simplest version of the model system, and first study that, and then slowly build it up. which is one way of tackling these problems. So we have been involved in a few studies where indeed immune cells, and checkpoint inhibitors have been shown to score very well in these organoids. And you can do it in a patient-specific fashion. Other labs have done a lot more. So there's a big activity now of adding tumor-associated fibroblasts, tumor-associated macrophages, T cells, other immune cells to organoids often in a personalized fashion. So you have to take the T cells from the same patient from which you grow the organoids. So that works. I mean, the oldest papers are 1.5 years, 2 years old. So this is a rapidly developing field. I think it will be important because you can actually rebuild the human situation rather than do this in mice. The blood vessels has been done a lot by companies. So the organ-on-chip companies who used to use cell lines, cancer cell lines, now are switching to organoids. And I think if you want to have blood vessels in a structured way grow in, you need the scaffold, you need the chip environment where you have separate compartments, one holding the cancer, another holding the endothelial cells, and then they actually beautifully create. We've worked a little bit with Mimetas in Leiden. They have beautiful images where you can actually then recreate blood vessels going into these tumors. And they've shown that you can now add immune cells and actually use the blood vessels and then exit and enter the tumor. So I think this can all be built, but it's, of course, this is going to be a lot of work to create something that really resembles a human tumor.
Thanks a lot.
There is a question in the fourth row on the right.
Thank you, Hans, for a great talk. I'm Valerie van Horen from Ona Therapeutics, a small biotech in Barcelona. And I had a very similar question because I'm also thinking how could we use these organoids for screening and understanding better our drugs. So apart from immune cells, could you somehow adapt your organoids to screen metastasis? So I think, what is the readout? So you have these organoids, you screen libraries to see, I guess you check for cell survival or proliferation. Can you check somehow if they have become less or more metastatic? Because I guess these organoids don't have a metastatic phenotype. Or if you would translate them again in mice, transplant them in mice, would you see then some metastatic readouts? Is that possible?
Yeah, so there's two answers here. First of all, there now but many other labs have sets of organoids where you have the primary tumor and a set of metastases from the same patient growing as organoids. I should say that also found by DNA sequencing by a number of labs, the difference between the metastasis and the tumor, and the original primary tumor, are not very large, if they exist at all. So we haven't really done anything to study metastatic behavior, but I know other labs have looked at motility, for instance, see if individual epithelial cells will leave the tumors. You might have noticed in some of the movies that I showed that actually we see when we have enough mutations, cancer mutations in a normal organoid. So if you make the cancers not from a cancer but starting from a normal organoid, they slowly, because they lose epithelial context, to become more invasive, and that would probably be a first stage towards metastasis. But I'm not aware of an assay that visualizes the metastatic process with organoids. But again, so you can have organoids from metastases. They're typically when in an advanced cancer patient, that is what we obtain as tissue and can grow. And for instance, for ovarian cancer, we often get both the primary tumor and all the metastases in the abdominal space because they remove them in a single surgical setting. Hope this answers your question.
Yes, thank you.
We have time for one more question. Tanya, front row on the left.
Now? Oh,
Yeah.
Fantastic. So Tanya Novobrantseva, and again, I think that everyone is fascinated by the talk. Thank you so much. But the question is, so you told us that organoids from pure sort of original cell type for the organ can form fascinating structures, right? And they are self-organizing, right? And you showed bile ducts that accumulate bile, etc. So my question is, do you actually see any differences in those structures compared to the actual organ? For example, there was beautiful work by Matthias Nerendorf a couple years ago saying that macrophages in the heart actually I needed to coordinate the beating of cardiomyocytes in the organ, right? So did you see any of the missing pieces, right, despite the obvious absence of other cells that are not formed by that original cell type of the organ and that are needing the presence of other cell types?
Yeah, so I should maybe say that we think we see the basic characteristics of an organ, of a tissue, but there's lots of modifications of those basic characteristics, which are often done by stroma, or like in the lung or in the heart. It's also muscle activity itself, so the stretching of the tissue will structure it. Of course, that can all be, again, mimicked with organs on chips, but is not present in the primary organ. So yeah, they are abstractions of the real organ, but they tend to harbor the essential functions in an experimentally attractive way. The moment we add other cell types, we see things changing. We see that there's some change morphology, so some functions now are controlled, whereas they normally are not, are just on or off and things like that. But yeah, again, you can create this complexity in a stepwise fashion, in a very controlled fashion. Yeah, but it's relevant. It's very relevant what you say.
Thank you.
Please join me in thanking Professor Clevers again for a wonderful presentation.